Good evening. This right here is the infamous Wuhan Institute of Virology. It's located right up the road from the Wuhan wet market, where, at least according to the official story, the first case of COVID was detected. And according to an exclusive report from Miss Natalie Winters over at the National Pulse, well, it turns out that the Wuhan Institute of Virology assembled a monkeypox virus genome, which allowed the virus to be identified through PCR tests. However, since there was no actual monkeypox in China, they used a certain method that put different fragments of the virus together in order to create it, which is a methodology that researchers have flagged for potentially being able to create a contagious pathogen. And even more interesting is the timing of the research. Because you see, the study that they released, you can see it up on screen for yourself, was published in February of 2022, which is several months before the latest international outbreak of monkeypox, which for your reference has now even reached the shores of the United States. And in their study, which was authored by 9 different researchers over at the Wuhan Institute of Virology, they appear to have identified a portion of the monkeypox virus genome, enabling PCR tests to be conducted in order to identify the virus. Here's specifically what these researchers wrote in their actual study. Quote, "Monkeypox viruses have strains that are more pathogenic and have been reported to infect humans in various parts of the world. Quantitative polymerase chain reaction, otherwise known as qPCR tests, is the gold standard for the detection of orthopox viruses, including the monkeypox viruses. For pan-orthopox viruses detection, the E9L DNA polymerase gene has been shown to be an excellent target for qPCR tests. For monkeypox virus detection, our colleagues reported that the C3L complement binding protein gene could be used as the qPCR target for the monkeypox virus Congo Basin strain." They then go on to state in their paper that within the borders of China, they did not have sufficient genetic information on this monkeypox virus for PCR detection. Here's specifically what they wrote, quote, "Since monkeypox virus's infection has never been associated with an outbreak in China, the viral genomic material required for qPCR detection is unavailable. In this report, we employed dual selective transformation-associated recombination, otherwise known as TAR, to assemble a 55 kilobit monkeypox virus's genomic fragment that encompasses E9L and C3L, 2 valuable qPCR targets for detecting monkeypox viruses or other orthopox viruses." Then, if you were wondering why they were doing this, the researchers actually go on to explain their reasoning for conducting this research and reassembling these virus fragments. Quote, "The primary purpose of assembling a fragment of the monkeypox virus genome is to provide a nucleotide template for monkeypox virus detection. As an efficient tool for assembling large DNA fragments up to 592 kilobytes in length, transformation-associated recombination assembly has become essential for preparing infectious clones of large DNA or RNA viruses." Now, if that sounds a little sketchy to you, in light of what we've lived through over the past 2.5 years, well, have no fear, because within this papers, the researchers acknowledged that this technique, which they were using, quote, "applied in virological research could also raise potential security concerns, especially when the assembled product contains a full set of genetic material that can be recovered into a contagious pathogen." Meaning that there is a real risk involved when you piece together fragments of a DNA or an RNA virus in order to create a monkeypox virus genome. However, as I mentioned a moment ago, you should have no fear, because the researchers, they went on to say this, quote, "In the study, although a full-length viral genome would be the ideal reference template for detecting monkeypox virus by qPCR, we only sought to assemble a 55 kilobit viral fragment, less than 1/3 of the monkeypox virus genome. This assembly product is a fail-safe by virtually eliminating any risk of recovering into an infectious virus while providing multiple qPCR targets for detecting monkeypox virus or other orthopox viruses." It's also worth mentioning that this study comes within the broader context of the Wuhan Institute of Virology conducting similar research into strains of bad coronaviruses that could also infect humans. Now, of course, I'm not saying that the strain of monkeypox that is spreading around the world has anything to do with this research. I'm only reporting to you the fact that several months before it began spreading, well, researchers over at the Wuhan Institute of Virology were using their research method to assemble different fragments of the monkeypox virus genome over in their laboratory. So make of that what you will. If you want to read this full study for yourself, it was published over in the Wuhan Institute's journal called Virologica Sinica. I'll throw a link to it. It'll be down there in the description box below, which is, of course, right next to that like button, as well as that subscribe button, both of which I hope you take a quick moment to smash. Now, while we're on the general topic of the Wuhan Institute of Virology, there's something else I believe that's worth mentioning, which is that within the latest edition of that same scientific journal, the Virologica Sinica, there appears another study showing that top researchers over at the Wuhan Institute of Virology, they appear to be, once again, engaged in gain-of-function research, this time by manipulating the H7N9 virus to be more lethal. Specifically, this study, which you can see up on screen for yourself, is titled, quote, Combined Insertion of Basic and Non-Basic Amino Acids at the Hemagglutinin Cleavage Site of Highly Pathogenic H7N9 Virus Promotes Replication and Pathogenicity in Chickens and Mice. And just as the name suggests, the study itself appears to outline a type of gain-of-function research using the H7N9 virus in chickens and mice. Specifically, the paper's abstract, it outlines how researchers, quote, generated 6 H7N9 viruses and then analyzed how those various amino acids altered the virus's virulence as well as its proclivity to infect. Here's what the study read, quote, we characterized the reconstituted viruses in terms of viral replication in avian and mammalian cells, thermostability and acid stability, cleavage efficiency, the virulence in mice, and pathogenicity and transmissibility in chickens. The I335V substitution of H7N9 virus-enhanced infectivity and transmission in chickens, suggesting that the combination of mutations and insertions of amino acids at the HACs promoted replication and pathogenicity in chickens and mice. Now, while the study appeared to only conduct research on mice and chickens, the researchers also noted that these H7N9 virus variants are, quote, highly transmissible in ferrets, posing the threats of pandemic potential in humans. And then, going further down into the actual results of their experiments, well, these researchers wrote that at least in one case study, the H7N9 variant caused a 35% mortality rate in mice, with the researchers noting that certain amino acids increased the virus's pathogenicity. Here's specifically what they wrote, quote, surprisingly, the monobasic mutants showed clear symptoms of infection, including shaggy hair and abdominal breathing after the second day of infection, followed by 15% of weight loss compared to the initial weight and death, with a mortality rate of 35%. In addition, each H7N9 virus can replicate efficiency in the lungs of the infected mice. Significantly increased viral titers in the lungs of mice compared to that of the control group, indicating that the insertion of the KRTA increased the pathogenicity of mice. Furthermore, if that wasn't evidence enough of gain of function, the researchers go on to allude to other changes that they made to the virus in order to test its increased lethality. Here's what they wrote, quote, notably, we observed that the insertion of non-basic amino acids at TA and IA in the H7N9 viruses caused death in mice despite having the same viral titer, which suggested that the HPAIV, H7N9, inserted with a non-basic amino acid posed a potential threat to humans. I guess that's what you call just another day over in Wuhan. If you'd like to read this study for yourself in its entirety, I'll throw a link to it. It'll be down in the description box below this video for you to check out, which is once again that description box right below that like button that I hope that you've already smashed. But if you haven't, take a moment to smash right now.